Hi everyone, welcome and good morning. My name is Layla Abul Hawa. I am the primary contact at Integral Molecular for RVP sales and technical support. Before we start, I want to cover a few housekeeping items. Attendees will be muted during the webinar. Please use the Q&A box at the bottom of your screen to type in any questions you have. Don't hesitate to type in questions as they come up. If we don't have a chance to answer your question, we will follow up with an answer by email. The webinar will be recorded and a link to the recording will be emailed to you. Now I'd like to introduce our team members who will be presenting today. We are joined by 3 integral molecular scientists, Haley Crawford, Kyle Doolin, and Joe Stafford. We are also joined by Sharon Willis, co-founder and VP of Sales and Customer Relations here at Integral. I will now hand it over to Haley Crawford to kick off our webinar. Haley?
Thanks, Layla. Good morning. I'm Haley Crawford, and I am the Virology Project Leader at Integral Molecular. We are a Philadelphia-based biotech company founded by virologists. We have 20 years of experience manufacturing virus-like particles and 11 years experience manufacturing reporter viruses. We have multiple platforms designed to study numerous viruses and the antibodies that target them, and we're going to tell you about them today. Next slide, Kyle. Back one, please. Thank you. We are proud to have developed relationships with our academic and industry partners, and the ever-increasing number of publications and patents is evidence of the broad usage and successful application of our various technologies. Next slide. Today, I will be presenting part 1. I will go over our comprehensive catalog of SARS-CoV-2 Reporter Virus Particles and how they are used to safely measure virus neutralization. I will touch on the updated FDA recommendations that guide the use of RVPs for development of vaccines and therapeutics against SARS-CoV-2 and the variants. And then Kyle Doolin, our project leader for molecular biology, will talk about the challenges for vaccines and therapeutics that Omicron poses, as well as our new offerings, new RVPs and antibodies. And then stick around for our live Q&A session at the end. Next slide. Our SARS-CoV-2 RVPs are fusion-competent pseudoviruses that incorporate the spike protein on their surface and are antigenically equivalent to live viruses. Their modified RNA genome renders them replication incompetent. They are capable of 1 round of infection of target cells and are therefore safe in a BSL-2 environment. The heterologous viral core contains a reporter gene that is expressed after membrane fusion, either luciferase or green fluorescent protein, and this enables detection of infected cells. Next slide. The readouts of the infectivity and neutralization assays are given as a percentage of infected cells when using GFP RVPs detected via flow or microscopy, as seen in the photo, or when using luciferase RVPs, relative light units are detected via a luminometer. We work in 384 well plates for high throughput assays, but many customers use 96 well plates. On the right is a neutralization assay of COVID-19 test serum samples. With your first order, we send you a free tube of HEC293 T cells that stably express ACE2. We also have VSV RVPs available as a negative control, and all of our optimized protocols are provided on our website. If you haven't downloaded these for a while, please ensure you get the latest versions today. And our scientists are also available to help you troubleshoot your assays. Next slide. Our SARS-2 RVPs have been shown to be comparable to the gold standard plaque reduction neutralization test, or PRINT, by many of our customers, and in particular at the University of Pittsburgh, shown here on the left. PRINT requires live virus in a BSL-3 lab it isn't high throughput and it's laborious, while our RVPs are safe, high throughput, and easy to use. If your goal is to obtain reproducible antibody or serum neutralization titers, RVPs are optimal for this. We measure the NT50 or neutralizing titer of our in-house antibody that neutralizes 50% of the RVPs. In the middle figure, we observed similar NT50 values for neutralization experiments performed on 5 independently produced lots of our SARS D614G variant RVPs. In the right figure, neutralization was performed using various volumes of SARS-2 RVPs from a single lot, between 5 and 40 microliters per well. And the curves and NT50 values were almost identical, showing the reproducibility and reliability of the RVPs. Next slide. Here are some recent exciting publications from our customers. Min Chen and their team at the University of Chicago created liposomal-based nanoparticles that express ACE2, and then used them to trap our RVPs. Here you can see the RVPs artificially colored green on the nanoparticle surface. Gordon Joyce and his team at Walter Reed performed high throughput neutralization assays with our RVPs on samples from animals that received ferritin nanoparticle vaccines. They showed that the vaccine elicited neutralizing activity an order of magnitude higher than that of the convalescent serum samples, shown in yellow. Next slide. Liang Chen and their team used 6 of our variant RVPs to test the neutralization titer specificity in a COVID patient given convalescent serum from another patient. At day 2, the titers declined over time, and then they increased as the patient produced endogenous neutralizing antibodies after 4 weeks. And our customers at PPD used our RVPs to validate their microneutralization assays, and they achieved precision, accuracy, dilutional linearity, selectivity, and specificity with our RVPs. Next slide. This map shows the clades of coronavirus sequences uploaded to nextstrain.org from the beginning of December 2021 until yesterday. Omicron in red dominates, followed by Delta in turquoise, and the new BA.2 strain of Omicron in orange is increasing in prevalence, as shown in the frequency chart at the bottom of the entire pandemic, and the virus is still evolving. Next slide. Oh, back one. Thank you. When a variant appears at a prevalence that is worth taking note of, or the WHO declares a mutant a variant of interest or concern, we produce, QC, and ship it to our customers as fast as possible. As of today, we have 70 SARS-CoV-2 variant RVPs available in our catalog in both reporters, GFP and luciferase. And we've also produced SARS-1 and MERS. We have an alanine scan library of SARS mutant RVPs, and we take custom requests. In addition, our catalog contains dengue, Zika, chikungunya, and VSV RVPs, and we are adding more viruses soon. Next slide. Our variant RVPs enable neutralization profiling for monoclonal antibodies or vaccine responses in patient or vaccinee sera. Our reference antibody on the left neutralizes the D614G variant in red, as well as alpha, beta, and gamma variants in blue. We tested a monoclonal antibody from a collaborator and saw weak neutralization of the beta variant in blue. Likewise, on the right, sera from a collaborator neutralized the D614G variant well in red, but not the 3 other variants. This profiling is essential for insights into vaccine responses and therapeutic antibody efficacy. The FDA recommendations for vaccine developers state that they should assess the immunogenicity of modified vaccines against variants, and that they can perform neutralization assays with wild-type SARS-CoV-2 virus or pseudoviruses. For developers of therapeutic antibodies, the FDA recommends that they implement a potency assay to confirm consistency and potency of each lot, They need to report the neutralizing activity against variants, and they may use pseudoviruses such as RVPs. Next slide. RVPs are not our only technology to support antibody development, informed by FDA guidances. Our shotgun mutagenesis epitope mapping platform helps to address the FDA recommendation that antibody developers should thoroughly characterize epitopes to identify escape mutations in circulating variants. Our epitope mapping rapidly maps antibody epitopes at single amino acid resolution. And for SARS-CoV-2 antibodies, results are available within 4 weeks. Our membrane proteome array can help address the FDA recommendation that preclinical studies should evaluate potential off-target binding of antibodies. Our MPA screens antibodies for off-target binding against 6,000 human membrane proteins. We will provide you with an IND-enabling report in approximately 4 weeks, and in many cases, this report is accepted by the FDA in place of tissue cross-reactivity studies. Next slide. Our customers at Immunome used our RVPs and our epitope mapping and our membrane proteome array for preclinical profiling of the anti-SARS-CoV-2 3 antibody cocktail. Using 10 of our RVP variants, they showed that the antibody cocktail neutralized these circulating variants from alpha through mu. We were able to map the residues in the epitopes for each antibody, and we found that they have unique and cryptic epitopes. And Immunome utilized our membrane proteome array platform to prove that their antibodies bound only to the target of interest. Immunome subsequently submitted an IND application to the FDA for this potential therapeutic. And I will now hand you over to Kyle.
Hello, everyone. Thank you, Haley. So we wanted to take a little step back and talk about the structure and the molecular biology of Omicron, since it is so distinct from strains that have come before. In some ways, Omicron has really rewritten the rules around what's happening in the spike protein, specifically for SARS-CoV-2. So Omicron, and in that I mean here B11529, though we will touch on additional variants that are emerging within the Omicron clade at the end. It has 37 mutations within the spike region. Compare this to the earlier mutants that had significant mutations of between 9 and 13, and you can see that it's just tremendously different. And these differences have a lot of consequences. Generally speaking, we can kind of classify these mutations as falling into 3 areas. There's those in the N-terminal domain, or NTD, shown in the middle figure, or the RBD, which mediates the interaction with ACE2, or within that S1, S2 interface that's shown kind of in that stem region at the bottom. Those in the NTD do tend to cluster around a neutralizing supersite around the 142 amino acid location. And so there were immediate concerns once it was sequenced that this area, this mutation would cause difficulties for vaccines and for antibody therapeutics. Even more mutated is the receptor binding domain, which is highlighted on the furthest to the right, where the receptor binding motif is those interfaces with ACE2, which is completely remodeled with many, many mutations. And this had really severe consequences for the therapeutic antibody space. Similarly, the mutations within the stem region have appeared before and are known to stabilize the S1, S2 interaction, further leading to the kind of rapid spread of Omicron, which we saw in the slides earlier. And so overall, what we see is a molecule that's changed dramatically, and dramatically at places that are very important for neutralization, either through vaccine or therapeutic antibodies. And this really needed to be investigated further. There have been some great studies on the structure with cryo-EM, in this case from the McCollum paper, which really looks at that interaction that Omicron has with ACE2. With so many mutations at that interface, how is it still able to bind to ACE2? And what we found was that it's a combination of mutations that increase binding, like S477N, creating a new hydrogen bond, the salt bridges formed by the Q to R mutations, and then the well-known N501Y with the pi stacking interaction that occurs there. All of these interactions work to compensate for an interaction that's not favorable in the K117 position. And so, and one of the very interesting things that they've discovered is that if you look at the way that these add together, it's not simply an additive process where, a little bit of affinity here and a little loss of affinity there adds up to where it's to some sum at the end, but they act in concert together. And in doing so, it retains binding at the same level as delta, which is an improvement over the affinity observed in the wild type. And so this leads to transmissibility like delta, but it also provides this immune evasion. And this interface is so different, and it showed later in the McCollum paper, that it even now allows interactions with other species such as mouse, which other variants hadn't been able to interact that SARS ACE2 interaction. So what we have is a completely new interface that needs to be disrupted by vaccines and therapeutic antibodies. And in a way, Omicron has evolved to disguise and hide from those sites that have been recognized in the past while retaining an interaction with ACE2 in a completely new way. And, as we tested that, here we're looking at the BioNTech paper that looked at the efficacy of the sera after various levels or dosing regimes. Shown on the left, here we're showing the titer in the sera as a dilution. So as at the bottom, you have very little neutralizing titer, and as you go up in the bar graph, you have an increased titer. And what we see is that after 2 doses, Omicron doesn't really, there's not very much protection against Omicron. In fact, 20 out of the 32 samples had are below that limit of detection and weren't able to find a titer. They followed up and has now been the custom in the public health with a 3rd dose. And after 1 month post that 3rd dose, there appears to be a higher level. But again, you can see a distribution that's lower than the others. So it's providing less protection than against beta and delta. And so this is the first direct evidence that these mutations that we were concerned about because of their locations had a real impact in the vaccine world. This has also been seen perhaps even more dramatically with individual antibodies that have been pushed forward for therapeutic treatment. Here, what you see is they're plotting the percent neutralization. So as you go up to 100%, you're getting more protection. And as you go from left to right, you're increasing the concentration of the antibody used. And what you see here is that Omicron in black is behaving very differently than the previous mutants. In particular, the Regeneron antibodies, both of them have significantly reduced neutralizing capability, as do the AstraZeneca molecules in the 2nd column and the Adagio molecules in the 3rd, and the Eli Lilly Abcellera molecules in the 4th. The unique outlier to that was Vir's sotrovimab or S309. It takes a little bit of a hit, depending upon the publication, that's somewhere between 2- and 3-fold, but it's still able to neutralize fairly well against Omicron, which we'll investigate a little bit more. But the consequence of kind of these sort of studies was that the emergency use authorization for the Eli Lilly and the Regeneron was removed. And so, this was a drug that had gone to market on an emergency use authorization, but then was pulled because of this inability to deal with Omicron and its rising prevalence. In a paper, they were able to co-crystallize or cryo-EM the S309 antibody, which is the parental sotrovimab, and how it interacts with ACE2. And what's really interesting is that there are these 2 residues right at the interface, the N440K and the G339D, that you may have predicted would interfere with the binding. And In particular, the N44K, the orientation of that lysine residue away from the interface was favorable to allow binding. And the G339D, here it's shown in the outward orientation, allowing binding. But in fact, we know that there are certain rotamers of this that will interfere and clash with this. And that's the structural basis for that reduction in affinity. And so what is really surprising is that even when we know what the epitope is, the consequences and the direct mechanisms of what that will do to the affinity and the neutralizing capability are not clear, especially in a remodeled ACE2 SARS-CoV-2 interface. As we started to work with Omicron and our RVPs, we immediately saw a big difference as well. So looking at the left panel here, we're looking at percent normalized infection. So as we increase in concentration, the amount of infected cells goes to 0 as we titrate in a neutralizing antibody. And we've been using VHH72 as our control, and it has done quite well against a large number of the named WHO strains, here shown with alpha and delta, completely neutralizing. And yet at our highest concentration of 100 micrograms per mil, we're only seeing partial neutralization for Omicron. And so this really raised a question for us about, what can we use as a control? We're not getting down to the, low single digit number of neutralization to see kind of that complete reaction occurring. And so on the right, we've plotted it against a number of the named strains, and Omicron. We can estimate that the IC50 is greater than 30. It's usually very difficult when you don't get a full curve to estimate it. By eye, it looks a lot closer to that 100 value, but we can certainly say, it's an order of magnitude higher than what we had seen in the past. And so at Integral Molecular, we have a membrane protein solution, or MPS antibody discovery platform, where we're able to generate antibodies against a wide number of difficult targets. Membrane proteins are our specialty, and viral proteins and viral membrane proteins, certainly a subset of that. And so we did an immunization campaign in chickens and pulled out antibodies by phage display. And in one campaign where we focused antibodies against the receptor binding domain, we found 25 antibodies and tested those against the Omicron strain and found that 10 out of the 25 were cross-reactive at a reasonable level, which is a pretty high cross-reactivity rate, and we think demonstrates the unique ability of chickens to provide diverse epitopes and antibodies. What you'll see here are 2 antibodies, and this is a flow cytometry analysis where in gray is the negative control, in darker gray is our positive control, the D614G mutant, or the kind of the parental, and then in Omicron in this orangey red, what the binding level is. And so here what you're looking at is that the maximal count, what the fluorescence is of the cells indicating binding. And so the G06 has a reduced binding to Omicron, but for F06, it seems to be as good, if not slightly better. And so I want you to remember kind of these profiles as we go and look directly at their neutralization. What we see for the neutralization, jumping right to the orange curves, what we're really interested in, how they do against Omicron, is that G06 has a reduced neutralization compared to Delta or the original parental. But F06 seems to have pretty comparable, though it is slightly decreased. And that's something that we'll come back to again, but in a similar ballpark because of that binding that we observed. Interestingly, it did not neutralize Delta, and that was something we also wanted to further recognize. But these 2 antibodies show neutralizing ability as good in the case of G06 and better in F06, getting down to single digit sort of percent infected, which will allow us to really estimate those IC50s and fully characterize the strain. We finally did our shotgun mutagenesis epitope mapping, and it really highlighted, along with the binding and the neutralization, what we were able to know about these antibodies. The G06 does have an epitope which was mutated in Omicron, and thus that's the basis for the reduction in neutralization. And F06 has no residues that are mutated in Omicron, and so should be able to equally recognize that. But it does have one mutation that would knock it out against Delta. And, one of the interesting things that we are, at this point speculating, but are hoping to look into further is that although it didn't have any mutations that were directly mutated in its hotspot kind of epitope map, perhaps because of that structural rearrangement, we're seeing something similar to what's occurring in the S309 antibody, right? And so, all of this kind of comes together to say that it's really important to test neutralization directly. That Omicron has really changed to evade vaccines and antibody therapeutics, and that unique binding interface with ACE2 and neutralizing antibodies really changes the nature of those protein-protein interactions. And that while we find binding and epitope mapping incredibly helpful in characterizing, it's not a direct predictor of neutralization. And it's very important that we directly measure neutralization if that's the property of either a vaccine or a therapy that we'd like to look at. And so, moving forward, we are committed to providing RVPs of all of the interesting strains moving forward. And at this point have the B11529 and BA2 came off this week and is ready for order. And we're monitoring subvariants of BA 1.1 and BA 0.3. But we'd also like to make available these antibodies as reagents for your own studies. And we're recommending specifically F06 as an Omicron neutralizing antibody. It's significantly better than some of the other controls that we've seen and reported Omicron neutralizers. And we felt it important to also provide the Delta neutralizer because it's a significant portion of the cases are still Delta. And so we wanted to make these available to the community and to all of you so that we can continue to learn more about Omicron and move forward in the post-Omicron world. And with that, we're happy to take questions.
Thank you, Kyle and Haley, for presenting today. My name is Sharon Willis, and I'm going to be moderating the Q&A portion of the webinar. I want to thank all of you for joining the webinar and for your trust in the reagents that we provide. We're happy and proud to continue contributing to the global fight against the pandemic. And we're scientists just like you, and we're always happy to help and to speak with you about your research and how our products and services can assist. I want to remind you, please put your questions using the Q&A button on the bottom of your screen. We did get a number of pre-webinar questions, so I will start with those. So the first question is for Kyle. Following up with what you just spoke about, Kyle, what is the amino acid variation across all the different variants of SARS-CoV-2? What does that landscape look like?
Yeah, we're certainly seeing some mutations pop up frequently. The D614G was immediate and almost universal. The N501Y that gave rise kind of the South African and the UK strains is being kind of permeated throughout, and the E484K mutation seems important. But Omicron really changed that. Before we were kind of picking and choosing and seeing those residues, and now we're seeing more divergence, and especially within the clade of Omicron, BA2 I think has 13. It is missing 13 of the mutations from wild type and has 8 additional ones. So the within that phylum, there's even higher diversity than we've seen in the past. But we're seeing mutations that we know their functions individually still cropping up. Because they provide this kind of biological function that's useful to the virus.
Great, thank you. This next question is for Joe Stafford, another one of our scientists who's joined for the Q&A. Joe, I know we sell GFP and luciferase RVPs, and the question is, do second and third generation progeny cells of the cells initially infected by RVPs continue to express luciferase and produce a luminescent signal?
Thank you, Sharon. Yes, the progeny cells will produce either GFP or luciferase, since the reporter gets integrated into the genome of the parental cells, so that should be passed down to the daughter cells as well.
Okay, great. So going to some more technical questions, Haley, what is the titer of the RVPs?
So we have tech data sheets available for every single strain and every lot we produce. On the tech data sheets for the GFP RVPs, we have the titer for every single lot, and that's infectious units per mil. And then for luciferase, because everyone has different luminometers and plate readers, we give the signal to background value. And also based on the titer and the signal to background, we give a recommended input for your plates.
Great. Joe, another question for you. Do we provide protocols for working with the RVPs?
Yes, all of our protocols are on our website, and Layla would give you the link to all the protocols that you need for both the infectivity assays and the neutralization assays. And then if you have any questions or problems with those assays or protocols, you can always email Layla, and she can get in touch with one of the scientists here, and we can help you and walk you through the assays.
Great, thank you. Another question for Kyle. How many spike proteins does a single RVP have? And what effect does this number have on the infection and the potential and neutralization?
Yeah, that's a really good question. I wish I had like a number to give. I think that's something we're investigating. But overall, the studies where we titrate in the amount of RVPs and find the same IC50 show that it's a low number, but it doesn't change the infectivity that much. So here we're changing the number, but not the concentration, per particle. Overall, it's probably a little bit on the low side, but it's relative to perhaps other sort of expressed proteins on the cell surface. But it appears very sufficient for looking at that viral entry event that we're interested in.
Great, thank you very much. Another question for Haley. Can you just remind us again what other RVPs that we sell outside of SARS-CoV-2, and then when those additional COVID-2 variants will be available and what we're working on?
So our catalog has all 4 serotypes of dengue. We've got Zika, chikungunya, VSV, SARS-1, MERS. We have Marburg and Ebola in the pipeline. And this morning, the Omicron BA.2 finished all its QC, and it'll be ready to ship. We ship on Mondays and Tuesdays. So if you get your order to Layla by Friday, you can have the BA.2 next week.
And then the BA 1.1 and 3 are on their way.
Those are in production.
Okay,
So
great.
approximately 4 to 6 weeks.
Awesome, thank you. A question for Layla. Is a license needed to use our RVPs?
Thank you, Sharon. That's a great question. No license is needed for the use of RVPs. However, reselling them is strictly prohibited.
Great, thank you, Layla.
You're welcome.
Joe, another question for you. Do you have any troubleshooting suggestions, or have we done any troubleshooting with regards to compromised samples, whole blood derivatives or hemolyzed serum that demonstrate false positive neutralizing reactivity in the RVP assay?
We do not have any data to this point right now, because we don't work with human serum a lot, but there could be ways that we could help you develop an assay to work around the hemolysis that you may be seeing that may be giving you a background. And we can work with you with our technical scientists here, develop an assay to to help you get rid of that background and actually determine the neutralization.
Great. All right, another question for Haley. Can the RVPs be thawed, aliquoted, and refrozen?
Yes, they can, but they are fragile. So we recommend 1 freeze thaw cycle. We sell them in 1 ml aliquots. So we have a 3 freeze thaw cycle on our website. If you thaw them according to the protocol, put them into smaller aliquots, and then refreeze in an ethanol dry ice bath, they should retain infectivity. But download the protocol for all the details.
Great, thank you. Another question for Kyle. Why target the spike protein? Are there other components of the virus that may be good targets as well?
Yeah, so there are some other proteins. None are nearly as large as the spike. The spike's a huge protein, and its exposed area, and it is the method for entry. We see entry as one of the key aspects to defense, and in fact, that's what the whole rest of the field has looked at. The possibility of neutralizing antibodies against others remain, but they really haven't been pushed in the clinic at this point. Though therapeutic intervention at other points in the virus life cycle, isn't an interesting question. It's just not one that we're particularly well suited with our RVPs to answer, though we certainly have that virology background and think about it quite a bit.
Great, thank you. All right, let's see. Here's another one for Joe. How stable, so we have an ACE2 stable cell line that we provide with our RVPs for your studies. How stable is the expression of ACE2 on our monoclonal cell line when passaging?
Thanks. The ACE2 expression should not decrease when grown under the pyrimycin selection that we recommend. We've taken these cells past passage 25, and we do not see any loss of ACE2 expression, but we recommend not taking them above passage 30.
Okay, great. Another one for Kyle. Omicron appears to have an altered preference of entry pathways favoring endocytosis. Would that affect the interpretation of results when comparing potency across mutants?
It's a good question. Let me think about it for a second. Yeah, I think it recapitulates most of the viral entry aspects. So I think that those features should still be present in our RVPs based on the pathway. It certainly might be cell type dependent. And so, there it may be more about your target cells than about the RVPs. A good question.
Yeah, there's lots to think about in this field of emerging variants. Okay, another question for Joe. Are the SARS-CoV-2 RVPs designed to work only with the HEC ACE2 stable cells, or will they also work with other naturally ACE2 expressing cell lines like Vero or Calu-3s?
That's a really good question. We offer the ACE2 cells because they highly express ACE2 on them. We do have collaborators who do work with the Vero cell lines and with the HUH7 cells, and they do show infectivity with our RVPs. But one of the things that we're seeing is there's so much genetic drift across multiple cell lines that some of the cell lines are not expressing ACE2 like is reported in the literature. So that's going to be very cell line dependent. So some labs are having really good luck, and others labs are not having good luck with the Vero cells or other endogenously expressing cell lines.
Okay, great, thank you. All right, another one for Kyle. Kyle, is incomplete neutralization common, 80 to 90%, as seen for S309 for some variants? And what do you think this means when the plateaus are not, when you're not reaching 100% neutralization?
Yeah. For therapeutic antibody, it's all about dosing, right? So what dosing can you get that's effective to knock down or neutralize? And so if we're not reaching that neutralizing titer, it comes down to how much can you force into a person in a dosing regime, or how quickly or frequently, and how long does it hang around? And certainly, half-life extension has been a hallmark of some of the therapeutic antibodies against SARS, because they want to keep those doses high over time. Its consequence for vaccines, I think, is a little bit unclear, especially since you see so much variability in a vaccine response. It's a little harder to estimate.
Okay, great. Thanks. Really great questions coming in, everyone. Thank you. Another question for Haley. Wondering how to obtain a significant signal to noise ratio, and what's the minimum amount, microliters, of Omicron RVPs that are needed to infect for a 384 well plate, for one well of a 384 well plate.
So we always recommend an input that gives you a signal to background or signal to noise of at least 200. The smallest volumes we've ever had for a 96 well plate are 1 or 2 microliters, but that's with SARS-1 or VSV. For any of our coronavirus variants, the lowest is 5 microliters, and that would translate to 1.25 for a 384 well plate. I think our Omicron current lot recommended input is 10 to 15. So approximately 2.5 microliters.
For a 384 well plate.
Could be good in a 384 well plate. Yeah, whatever we recommend for a 96 on the tech data sheet, you just divide by 4, and that's the equivalent. So I'd say between 2.5 and 5 microliters. But check out the tech data sheet for the specifics of each lot.
Right, great. Haley, while I have you, can you just review again the antibodies that we now have for sale, and what's their neutralization profile?
So we have the integral neutralizing antibody G06 that strongly neutralizes Delta, weakly neutralizes Omicron, and then we have our F06, which neutralizes Omicron. That's our Omicron neutralizer. And does it? Oh, it doesn't. No, yeah, sorry. It doesn't neutralize Delta, and it does neutralize the parent strain D614G. So G06 is the Delta neutralizer, F06 is the Omicron neutralizer.
Great, thank you, Haley. It's a lot of numbers to keep track of in letters. Joe, question for you. Do you recommend one reporter over the other, GFP or luciferase?
So that comes down to what you want to measure. Some labs are not equipped to run flow cytometry, so we would tend to lean them towards luciferase assays. There's a difference where if you're using GFP, you can do individual cell interrogation to see the actual percentages of cells that are GFP positive compared to the whole population of cells. Whereas luciferase, it's more of a population-based luciferase signal, because you're reading the whole well and not individual cells. So it's going to depend on what your lab is set up for. Both of the reporters that we use are very good, and it just depends on what equipment you have in your lab.
Great, thanks, Joe. Okay, a question for Haley. You talked about the FDA, and you're part of the talk. Does the FDA accept RVP data for regulatory submissions?
Yes, for vaccine or antibody developers, the FDA guidances say that you can use live virus or pseudoviruses. So RVPs are very suitable.
Great, thank you. All right, another question has come in for Kyle. Any plans to produce anything with any particles with S protein WIV-1 or other beta coronavirus S protein particles?
Yes. What can I say? We as a custom order, if you're interested in other coronaviruses, that's certainly something that we've been able to do in the past. And there's additional interest now in the beta coronaviruses. So there's some similarity. There's also quite a bit of difference, but get in touch with Layla, and we'd love to talk to you about your project.
Great. All right, I think we have time for a couple more questions. One more for Joe. What plates do you recommend for infectivity and neutralization assays?
If you read our protocols online, they have the recommended reagents and recommended plates that we use for each of our assays. We just updated most of the protocols in January. So just please visit our customer documents page for the latest versions, and they'll tell you exactly which plates we use. And if you cannot find those plates, you can always email Layla, and we can sometimes suggest alternative plates.
Great, thank you. That is the end of the questions that have been coming in. So I want to thank everybody again for joining us today. Thank you to our panelists, Kyle, Haley, Layla, and Joe for presenting today. And Kyle, can you go to the thank you slide? So thank you. Here we are at Integral Molecular in sunny times. Again, we're working hard along with you to fight this pandemic, and we really look forward to our continued work with you. And please don't hesitate to reach out to Layla if you have any questions or need anything. So thank you, everyone, and I hope you enjoy the rest of your day.